I'll try to keep it like a super lightning talk. Um, okay, I'm Ananthan, and I'm going to be presenting on MC-Funcformer, a foundational model of microbial community metabolism. So this is our motivation for the study. Say you're studying the gut microbiome, and you do the typical thing of getting the microbial abundances, and you feed it through some sort of machine learning model to predict phenotypes. But turns out you're not only interested in the gut microbiome, you're also interested in the soil microbiome. So you can't really use the abundances with your existing machine learning model to understand both the soil and the gut microbiome. So the workaround is that people do is using functional representations of microbial communities instead. So you pretend that each microbe is a bag of genes, and you use a tool like PyCrust to convert your microbial community from microbes to a bag of genes, which you can then get a functional abundance vector from. We take it one step further, and we rank these functional abundances to get a sequence of genes. And when I say genes, I actually mean gene families or KOs. It's just easier to write down gene than gene family. Okay, so the reason why we do this is so that we can then use ideas from natural language processing to then understand microbial communities. So we have our model MC-Funcformer, which stands for Microbial Community Functional Information Transformer. And so the idea that we do is we use the idea from mass language modeling, which basically just means you hide certain parts of your sequence. In this case, it's a gene family, and you ask the model to predict it. And the intuition behind why this should work is if a model was to correctly predict which gene should occur in a particular position, it should take into account the context of the entire sequence and figure out which gene would make more sense at a particular abundance level in that microbial community. And that will force the model to learn some sort of biological information. And once we pre-train our model, the goal is to then use the vectors that it has learned for downstream tasks, including predicting host phenotypes, predicting environmental properties, and getting contextual embeddings of these gene families. Some data-related stuff that we did, we removed gene families that occur in less than 1% of samples. There's actually a part B that somehow disappeared, but we also removed genes that have low coefficient of variation. And our model was trained on about 170,000 microbial communities that we got from Cheetah. We're currently working on training a model that has even more data with 2 million samples that we're getting from the Microbiome Atlas project, MAP. But yeah, so, okay, so first, a sanity check. Do MC-Funcformer embeddings actually capture biologically relevant information? So what you see here is a visualization with labels from the Earth Microbiome Project, and it passes the first sanity check. Free living versus host associated are pretty clearly separated, although you do see a big island of cells or island of points here, which are a mix of free living and host associated. So we want to dig into that a little bit more. At the higher resolution, we see that it's a mix of plant and non-saline. Kind of interesting, but don't really know what that means yet. So you go at the highest resolution of annotations from the Earth Microbiome Project, and turns out it's a mix of soil microbiomes and plant rhizosphere. So it makes sense that they're there together. Okay, so that visualization was fun and all, but does MC-Funcformer actually capture information more than just looking at the relative abundance of these functional families? So to test that out, what we did was we did some k-means clustering on the embeddings and the relative abundance vectors, and calculated the adjusted mutual information between the clusters and the labels from Earth Microbiome Project. And turns out that MC-Funcformer embeddings consistently across all 3 levels of resolution did capture more information about the true labels than the relative abundances. Okay, but the real thing that we want to do is to predict the properties of microbial communities using the embeddings from MC-Funcformer. So we did that with, initially we did it with diets. And here what you see is comparing using the embeddings from MC-Funcformer to, in blue, using the abundance vectors from just from PyCrest. And it seems that consistently using the embeddings from MC-Funcformer gives you higher performance than using the raw data. We did the same thing with diseases associated with the gut microbiome. And again, MC-Funcformer embeddings did better than the raw data. And finally, we also try to predict environmental variables from soil and water microbiomes. And for 3 out of the 4 test cases that we tried out, using MC-Funcformer did better than just using the raw data. Okay, so next, we want to look under the hood of the model for, in this case, just one particular gut sample. So the first thing that we did was we wanted to see how mass language modeling actually worked with our sample. So when you do mass language modeling, you can also calculate an accuracy score for how well the model predicts the masked tokens. And what we saw was that the model was significantly better at predicting tokens that were more abundant than those that were less abundant. And that makes sense because the less abundant ones are more controlled by noise, and you can't expect the model to be able to get through all that noise. So this made sense to us. And finally, what we also wanted to do was to look under the hood of the model for, this is for the same gut sample, and see what the attention layers of the model actually learn. And the attention layers just tell you what the model looks at when trying to make a prediction for any particular token. And what we see is that for the first 3 layers, there isn't any clear pattern. By the 4th layer, there are 3 bands, which means that the model not only looks at tokens that are of a similar abundance to the token that you're looking at, but it also looks at what the most and least abundant tokens were in that community. And by the last layer, there seems to be some sort of local information because we started seeing these squares that were formed, which kind of tells us that there are these clusters of co-occurring genes that the model is trying to catch at. But yeah, so in summary, MC-Funcformer provides a functional representation of microbiomes that is generalizable to different kinds. These representations tend to be more informative than gene family abundance vectors. And these representations can be used to predict the phenotypes of microbiomes or their host. Yeah, and so I'd like to thank Zihan, Malcolm, Veronica, and Sergey, who are co-authors on this paper. 3 of us are from University of Illinois. Veronica is from Gladstone.
Thank you so much.